Apoptotic stimuli damage mitochondria. The damaged mitochondria release several proteins like cytochrome C or DATP, SMAC or Diablo, AIF, HTRA2 or OMI, and ENDOG. These proteins stimulate apoptosis. Let us see how cytochrome C or DATP stimulates apoptosis. Cytochrome C or DATP released from mitochondria activates APAF1. Active APAF1 then activates caspase 9. Caspase 9 is the initiator protein. The active caspase 9 then activates caspase 3 and caspase 7. Caspase 3 and caspase 7 are effector proteins. Active caspase 3 and caspase 7 activate cellular targets like proteins of nuclear lamina and proteins of cytoskeleton. Active cellular targets then activate apoptosis. In mammals, BCL2 protein prevents release of cytochrome C or DATP from mitochondria. As a result, cytochrome C or DATP cannot activate APAF1. Inactive APAF1 does not activate caspase 9. Inactive caspase 9 does not activate caspase 3 and caspase 7. Inactive caspase 3 and caspase 7 do not activate cellular targets. Inactive cellular targets do not activate apoptosis. Mammals have two BH3 only proteins named as BEAD and BEAM. BEAD and BEAM inhibit BCL2. So, inhibited BCL2 no longer inhibits the release of cytochrome C or DATP from damaged mitochondria. So, cytochrome C or DATP is released from mitochondria. Cytochrome C or DATP then activates APAF1. Active APAF1 then activates caspase 9. Caspase 9 then activates caspase 3 and caspase 7. Active caspase 3 and caspase 7 then activate cellular targets. Active cellular targets then stimulate or activate apoptosis. IAPS block activation of caspase 3 and caspase 7 by caspase 9. So, inactive caspase 3 and caspase 7 do not activate cellular targets. So, inactive cellular targets do not activate apoptosis. So, cell survives. IAPS also block the activation of cellular targets by caspase 3 and caspase 7. As a result, inactive cellular targets do not activate apoptosis. As a result, cell survives. Now we will discuss about how SMAC or Diablo stimulate apoptosis. SMAC or Diablo released from mitochondria bind to IAPS in the cytosol. This binding blocks IAPS. Blocked IAPS do not bind caspase 3 or caspase 7. So, blocked IAPS do not block activation of cellular targets by active caspase 3 and caspase 7. So, active caspase 3 and caspase 7 activate cellular targets. Active cellular targets then activate apoptosis. Previously in this video, it was said that IAPS block the activation of caspase 3 and caspase 7 by caspase 9. So, inactive caspase 3 and caspase 7 do not activate cellular targets. So, inactive cellular targets do not activate apoptosis. After release of SMARC or Diablo from the damaged mitochondria, SMARC or Diablo blocks IAPS. Blocked IAPS do not bind to caspase 9. So, blocked IAPS do not block the activation of caspase 3 and caspase 7 by caspase 9. So, active caspase 9 activates caspase 3 and caspase 7. Active caspase 3 and caspase 7 then activate cellular targets. Active cellular targets then activate apoptosis. BCL2 also blocks the release of SMARC or Diablo from damaged mitochondria. As a result, SMARC or Diablo does not block IAPS. The IAPS then block the activation of caspase 3 and caspase 7 by active caspase 9 and also block the activation of cellular targets by active caspase 3 and active caspase 7. So, cellular targets remain inactive. Inactive cellular targets do not activate apoptosis. BEAD and BEAM block BCL2 protein. Blocked BCL2 protein does not block the release of SMARC or Diablo from the damaged mitochondria. The released SMARC or Diablo then inhibits IAPS. Inhibited IAPS do not block the activation of caspase 3 and caspase 7 by active caspase 9 and also do not block the activation of cellular targets by active caspase 3 and active caspase 7. So, active cellular targets activate apoptosis. The proteins like AIF, HTRA2 or OMI serine protease and endo-G help to kill the cell. HTRA2 or OMI causes cleavage of IAPS. So, IAPS no longer inhibit the activation of caspase 3 and caspase 7 by active caspase 9 and also no longer inhibit the activation of cellular targets by active caspase 3 and active caspase 7. As a result, active cellular targets activate apoptosis. AIF is a flavoprotein. Normally, AIF acts as a NADH oxidase. In presence of proteases, AIF undergoes cleavage to produce cleaved AIF. The cleaved AIF then moves to the nucleus. In the nucleus, the cleaved AIF causes chromosome condensation and DNA fragmentation. As a result, apoptosis occurs. The effects of AIF, HTRA2 or OMI and endo-G are caspase independent. So, we can conclude that all apoptosis do not involve caspases. Now, some important points. The first mammalian apoptotic gene to be cloned was BCL2. The BCL2 gene was isolated from human follicular lymphomas. Humans have 15 different caspases. Mammalian BCL2 protein is 23% homologous to CED9 protein of nematodes. Both BCL2 protein and CED9 protein contain one transmembrane domain. Both BCL2 protein and CED9 protein are present in outer mitochondrial membrane, nuclear membrane and ER membrane. In outer mitochondrial membrane, nuclear membrane and ER membrane, BCL2 protein and CED9 protein act as sensors that control the apoptotic pathway in response to external stimuli. Please like, subscribe and share.